It's very good to be back. We've spent a lot of time here at JGI working towards doing whole genome association work in Poplar. And I'm going to present where we are to date. The accomplishments that I'm going to be presenting really represent the contributions of dozens and dozens of people, and most of them are listed here. And I'm afraid I won't take much time pointing out the individual contributions, but recognize that there was a whole team of people that worked to get to where we are today. For those of you that don't know about Poplar, and I can't believe any of you don't know about Poplar, but if there are some of you out there, Poplar is an undomesticated tree. It's dioecious. It's one of the oldest living organisms on the planet. There's estimates that individual genets can live up to be 10,000 years old. It also represents one of the largest organisms on the planet. A single clone of Poplar occupies 110 acres, a single gene in Utah. And so these are very interesting organisms. They're one of the fastest growing temperate tree species in the world. And as a result of those characteristics and others, it's one of the model organisms for bioenergy, for the conversion of lignocellulosic feedstocks to transportation fuels. The specific objectives of the project are to identify genes that are controlling cell wall biosynthesis. And so it doesn't take long. You can go to the literature and look up cell wall biosynthesis, and you can come up with a handful of candidate genes that are known to affect lignin, cellulose, and hemicellulose biosynthesis. But we're taking an a priori approach to this with no preconceived notion of what is controlling cell wall biosynthesis. And so a whole genome or genome-wide association study is the direction we're going. To do that, though, it requires three basic bits of information. You need the appropriate population so that there is high power, high ability to detect associations. You need high quality phenotypic data with low standard errors on measures of the phenotype. And you need informative genotypic data. And so for the rest of the talk, what I'm going to present is how we were able to assemble a population, how we were able to derive appropriate phenotypes, and how the genetics and genomics and resequencing data has helped us inform our association tests. I will step through those individually. Sorry about the reverb. So let's start out with the appropriate population. As I mentioned a moment ago, Populus is a non-domesticated, wild, perennial woody plant. It exists, Populus trichocarpa exists along the Pacific coast. And we made a collection of 1,400 genotypes from the wild. And you can see the locations here. And those individuals then were brought into several common gardens. Because natural selection operates upon various gradients, whether it's temperature, moisture, photoperiod, we knew that there was going to be genetic structure within the population. So the first thing we did was try to examine or tease apart whether or not we had a panmictic population in our association test. And so we used microsatellites initially. We had probably about 500 microsatellites that we knew were segregating in this population. And we did a standard population genetics test. And based on this, you can see from this distribution, this is one of the microsatellites, that we did have structure within our population. And when you looked at expected heterozygosity or FIS values, it became apparent that there was a difference between the northern region and the southern region depicted by that gold line right there. So in fact, we threw out 300 of our original collections that occurred north of that boundary right there based on microsatellite data. So our initial population included now 1,100 individuals. Interestingly, when we began to look at the data based on microsatellites, we came up with two hypotheses which would account for this structure. One would be that there were island populations that recolonized the area after the last glaciers receded. Alternatively, you could imagine that there was integration from an alternate species. The orange is Populus balsamifera, the green is Populus trichocarpa. They're sympatric over some of the northern portions of Populus trichocarpa. So we didn't know which of these two phenomenon were accounting for the structure, but we knew that that structure would create an artifact in the association study. So we eliminated any of the genotypes north of that boundary right there. I'll get back to this in a moment, but let's move on. Those 1,100 individuals were then clonally replicated and established at 4 common gardens. At each common garden, they were planted in a randomized complete block design, and there were 3 blocks in each of the 4 environments. We selected these 4 environments designated here by the gold stars, one along the riparian zone of the Fraser River here in British Columbia, another coastal site at Clatskanie, Oregon, an interior riparian site at Corvallis, and a high elevation drier site at Placerville, California. So by establishing these common gardens, it gives us the ability to separate genotype by environmental effect on the association test that we're conducting. This is what the trees looked like at the end. I don't know if you can see that very well, but at the end of the first year, we had quite a bit of variability in tree performance. They've now completed their 2nd year of growth, and they're entering their 3rd year of growth. And I'll present some data from the year 2 wood chemistry results. Simultaneously with that, we developed a series of high-throughput phenotyping assays. And because we were looking at cell wall formation and ultimately the production of biofuels, we developed a series of screens that allowed us to measure either cell chemistry or performance in co-cultivation with enzymes and bacteria. And based on this, we have a series of phenotypes that we were using in the association work. We have a pretreatment assay, which is based on high-pressure acid and steam treatments, a sugar release assay, which is based on a cocktail of enzymes, and then a chemical assay based on IR and MBMS, molecular beam mass spectrometry. So those were the phenotypes. When we applied those assays to the population, we saw that the distributions of phenotypes were fairly normal. This is all 1,100 individuals for S to G ratio. S to G ratio is syringeal to guaiacil forms of lignin. They're the precursors, the monomers that go in to form the polymer, and they provide the covalent structure for lignin biosynthesis. This is composition of lignin, percent lignin, and again we see a wide range of phenotypic distributions for those traits. That is true also for xylose, glucose, mannose, all the carbohydrates found in the cell wall. I'm just showing the data here for lignin. In addition to the normal distribution, we found that there were some extreme phenotypes, and they're designated by these red boxes. Those red boxes allowed us then to tease apart genetic controls for those extreme phenotypes. I want to mention here that there was a slight, even though we threw out the 400 or 300 individuals in the northernmost portion of the distribution, there was still a slight negative correlation with latitude, such that lignin increased as you went further north. So we still had some indication that latitude was having an effect on some of the phenotypes we were measuring. When we looked at those extreme phenotypes, there was a negative association between sugar release, glucose content, xylose content, and all the measures of lignin. So there was a negative correlation between lignin, polyphenolic, and sugars found in the cell wall. What you see here again are all 1,100 individuals for percent lignin content and S to G ratios in lignin. And we selected these based on an orthogonal comparison and then looked at sugar release. And this is a very complicated graph. It's probably hard to read from the back. But the bottom line is that these are pretreatment conditions. The column on the left is the most severe pretreatment. The column on the right is the least severe pretreatment. What we see here is 5-carbon sugar xylose, glucose, and total sugar release under a pretreatment condition. And the slopes of the lines, the red and black lines, indicate the release of sugar under S to G ratios above 2 for the black lines and below 2 for the red lines. And this was the first time that we were able to demonstrate that you could uncouple the relationship between syringeal and guaiacil forms of lignin and percent lignin content and have an impact on sugar release. So the point being here is that the phenotypic values that we're measuring in the association study allow us to uncouple these very complex biochemical traits. And so we're getting clean, independent measures of each of the phenotypes with a high degree of precision. Simultaneously then with all of that, we were also resequencing genotypes, and this work was done here at the JGI. And there were 19 genotypes that were included in the resequencing effort to an average depth of 30x. And so this was a table indicating the number of SNPs that were detected per genotype. And some of these had not been completed yet, but we've now completed that. Those 19 came from these positions that we see in the red circles. In addition to those 19 genotypes, we also did deep RNA sequencing to determine SNP frequency in the RNA libraries. And so we had pairwise comparisons of both resequenced whole genome SNP determination and SNP determination from RNA-seq. There was a high correlation of matching between each of the 19 genotypes and the reference genome. And this really reflects the bioinformatics work that was done here at JGI to identify the highly informative, high-confidence SNPs. In total, there were 14 million SNPs that we ultimately brought into the whole genome array association study. But in total, there was probably somewhere around 54 million. So we didn't look at all the SNPs, and we didn't interrogate all the SNPs. We used filters based on their confidence, based on their positions, to then propagate those into the association study. Once we had resequenced those genotypes, we had to characterize linkage disequilibrium. And what you see here is a graph of LD across the 19 chromosomes. So that's chromosome 1, 2, 3, 4, all the way down to chromosome 19. And there are a couple of things that stand out here. LD varies greatly across the genome, as you'd anticipate, but it varies by a factor of 300. And so we have 2,000 high recombination positions within the genome. So this is a depiction of the historic recombination rate based on the resequencing information from those 19 genotypes. The effective population size here is about 5,000, and we have about 2,000 of these very high recombination areas. When you look across the genome, there's a 5 order of magnitude variance in the recombination rate in a 50 locus sliding window. So depending on where you are, recombination rates could be 5,000 times higher than what you see just in the neighboring region. These also vary among genes, and so if you just look at coding regions within the genome, the variability increases to an 8 order of magnitude increase. When you lay estimates of recombination frequency based on genetic mapping on top of the estimates of recombination frequency from resequencing, you get very good depiction or representation. So the recombination rate, that's for linkage group 7. The black line is based on the genetic map for one pedigree, and the red line is the historic recombination rate. And so you begin to see that these rates of recombination are controlled and persistent in your population. So these aren't just artifacts of individual genotypes coming together and being crossed to create a progeny set, but in fact are controlled by the historic evolution of the population at the genome level. A couple of other things. The amount of recombination was negatively correlated with methylation, significantly negatively correlated with methylation. It was significantly negatively correlated with repetitive elements. And so based on this information, then we developed a multi-regression equation to predict recombination at any position in the genome. With the addition of the GC content and CPG islands, we could predict 80% of the recombination rate at any position in the genome. So that then gave us the power or the ability to determine linkage disequilibrium at any position along the chromosome. Another thing to note is I did mention that there were roughly 2,000 hotspots that had a Bayesian factor above 300, so highly significant recombination positions within the genome. And these typically occurred at 3 to 4 kb upstream of the genes themselves. And in fact, 10% of those 2,000 share a very specific non-annotated sequence of DNA. So we still don't know what that sequence is doing, but it's common. It's found among 10% of those high recombination sites, and we're trying to investigate the role of that conserved motif. Then if we look at the decay, linkage disequilibrium by latitude. So how are the populations structured? And these are the independent individual samples or locations across latitudes, and that's the R squared or LD values across the sampled population. There is primary variation associated with latitude. 17% of the variation in LD is controlled or associated with latitude. But if you exclude these extremes here and this extreme here and what we're referring to as the core range, those genotypes central to the population, you get a very linear relationship. And so you can use this information both at the range-wide level and at the core level then to tease apart what's happening in terms of associations between individual genes at individual positions in the genome and the associated phenotype. We wanted to know why this was occurring. We thought we had scoured out of the population the structure that was associated with latitude. And when we looked at the resequencing data, it turned out that there were 8 genes related to photoperiod reception that account for the majority of that clinal variation. And when we looked at those genes, these are phytochromes and other genes associated with light detection. When we looked at LD estimates across the genome, and I apologize if you can't see, this is for phytochrome and other light receptors, you see that the LD is much smaller. And so it appears that there was selective sweep that occurred in the population when Populus trichocarpa recolonized the Pacific Northwest after the last glacial event, and that selective sweep was based on photoperiod response. If you then look at the genome-wide analysis, which is this line here, and you can see that LD decays at a slower rate, and so that's this graph depicted down here. And what this then does is allow us the opportunity to do association studies on a range-wide basis or on the core population basis. And this difference is significant in terms of detecting significant associations. The other thing to note here was that there was no substantial difference in the technology, the DNA sequencing technology, for our estimates of LD. And this tells us that the original efforts computationally to identify highly informative SNPs was effective, and such that either Illumina sequencing or Sanger sequencing gave us similar estimates. So we're fairly confident that there isn't hidden sequencing error or read error contained in this data set. So those are all important criteria then for doing association work. I wanted to get back to this story. Remember I told you that we couldn't decipher based on SSRs whether it was hybridization or recolonization. But based on whole genome resequencing, we're now fairly confident that this represents a colonization event from an island population, probably somewhere in California, and that all of Trichocarpa has recolonized the Pacific Northwest from that population. And we weren't able to do that based on SSRs. It was only once we looked at the whole genome resequencing data and looked at variability, recombination rates, and linkage disequilibrium across latitudes that we were able to then determine how this population that we've sampled actually came to be. Now on to association work. Our goal, and we're driving towards whole genome genome-wide association studies, but we wanted to determine whether or not we had a robust population, whether or not our phenotypic data was reliable, and whether or not the resequencing data was reliable. So we undertook an Illumina Affinium bead array, and so we created a bead array based on the resequencing data. But to do that, you have a finite set of targets that you can sample. And so to determine what targets went on the bead array in order to determine whether or not association genetics was going to be effective in this species, in this population, we had to narrow our candidate gene list down. So we used QTL analysis. QTL analysis identifies regions of the genome that contain genes associated with phenotypes. We use the same phenotypes that I've mentioned already. And the problem is that those intervals are fairly large. And most of the genes in those intervals are just computational representations of gene function. Very few of them have been functionally validated. On top of that, over half of them in the intervals tended to be unknown, putative, or hypothetical genes. So we then laid on top of that microarray data. So based on 179 QTL intervals, there were 49 that co-located to common loci. There's some examples of those QTL loci here. 27 of those 49 affected multiple phenotypes, lignin, sugar release, etc. We came up with a candidate list of 2,583 genes that went onto the bead array. We then took the SNP data that was from the 19 resequenced individuals and then spotted a total of 38,000 SNPs on the bead array. Because of linkage disequilibrium, that actually represented about 90,000 positions within the genome. We ran that, and of the designed 38,000 SNPs on the array, 34,000 were successful. This was technically successful. We got data back from them. This is an example of what the data looks like. We excluded any genotypes that had 20% failure rates, and we excluded SNPs with no calls 10% of the time. So in total, we had a population of SNPs around 32,000 segregating SNPs or variable loci within the population. That was then applied to the phenotypic data that I mentioned. Sugar release, lignin content, S to G ratio. We used a mixed linear model with a kinship criteria for a covariate, and our significance based on a false discovery rate had to be below e to -7. And what you're seeing here are the genes associated that we were able to detect using the bead array and this population, the phenotypes. So this is all 1,000 individuals. And so what you see here are a list of phenotypes, the genes associated with those phenotypes, their probabilities, e to the -23, -24, -27, highly, highly significant. And this is the percent phenotype that's explained by these genes and the SNPs associated with those genes. 17%, 18%, 20% of the variation in this population is attributed to these genes. So here we have an indication that the population structure is correct. We've been able to tease apart or eliminate spurious variation due to latitude, that it does have enough genetic variation to be a robust test. Our phenotypes appear to have low standard errors, so we're getting good phenotypic measures, and the statistics behind the association study look very, very promising. I should note here that when we took those 2,500 genes from the QTL intervals that also had microarray or expression data with them, we looked at SNPs not only in the coding regions, but in the 5' and 3' UTRs, and we included SNPs that were within a 5 kb window. Because I mentioned earlier that our LD analysis indicated that some of these recombination hotspots were 3 to 4 kb upstream of the gene itself. So many of these SNPs are not in the actual structural coding region, but are adjacent to the coding region. That's very, very similar to what we see in most of the SNP studies in humans. The exception to that are corn and rice, where you do see SNP variants in structural genes. Here in poplar, most of these occur outside the coding region. So they're associated with some regulatory element. What's next? We're going to complete the estimates of LD and write that manuscript up and submit it. We're also completing the association map based on the Illumina bead array. The field trials will continue up through 2012. We'll continue taking phenotypic data. We're trying to measure every phenotype we can think of, in addition to just the wood chemistry and sugar release and pretreatment assays. We're also measuring performance, height, diameter, disease, insect resistance, etc. We're in the process of resequencing that core 800 genotypes. We're going to evaluate that at each step to make sure that the information is informative, but we're up through, I think we're up through 80 of those genotypes now have been completely resequenced, and we're going to attempt a genome-wide association study in 2012. I think with that, I'll answer any questions, and
questions for Gerry? Please wait until you have a microphone in hand.
You've gotten some great resources together and making some great progress, but I had a question on the flowering time ones. So when we do look at flowering time genes, they almost always show more LD because they don't have the differentiated, and they don't have an opportunity to recombine. So why do you think you're seeing less LD at the photoperiod genes?
You know, that's an interesting question. This is a perennial organism, and so it goes through a juvenile mature transition. The ability to reproduce is determined by photoperiod as well. And so we think that it has something to do with maturity as it moves forward. It is counterintuitive. The results surprised us. We don't have a clear explanation other than we think that it has to do with the maturity of individuals as they moved further north and photoperiods changed, flowering time changed. And so it was an intriguing outcome. I'm not sure we have a clear explanation, but it's something we're looking into.
Gerry, do you think your GWA is landing in non-coding regions versus the genes is because it's gradual selection versus real domestication selection?
That's a great point, Eddie. The population has not gone under any domesticated process. This is native materials that have been selected only by their environment. And because we're not looking at or dealing with populations that have been bottlenecked through domestication, it may be associated with that.
I have a question about the hotspots here, back in this. You know, these hotspot estimates are very, very sensitive to various kinds of sequencing errors. So, you know, if the SNPs you're looking at actually are further apart than you think they are, for instance, there's some bit of DNA between them, or so how confident are you that these really are hotspots, right?
So the reference genome, of course, determines a lot of your ability to detect SNPs, and we're continually improving the reference genome. And there are certainly gaps and errors in the current assembly. Outside of that, though, as we resequenced, that was all based on Sanger sequencing and the original version 2 assembly. The resequencing was done by Illumina, and we certainly had reads that fell outside of the reference genome, possibly in gaps or our sequencing errors. We filtered all of those out. We also looked at only those SNPs that were shared by position across 95% of the resequenced genotypes. And so for a SNP to move into that 14 million category that you saw just in the one slide, it had to meet several criteria. It had to be in a position where we had high confidence. It had to be in a position with physical structure adjacent to a certain distance away from other SNPs. And so based on the performance of the bead array, and by the way, Illumina will only guarantee 85% success on their design. We were hitting 97% success on the bead arrays. I'm fairly confident that we have eliminated most of the read and sequencing error based on the computational approaches we took.
Right. I mean, I'm not really questioning the SNP calls. The question is how much is in between the SNPs, right? So I think it's very important when you try to estimate, you know, LD hat or whatever you do, to make sure that these SNPs actually are separated by the amount of DNA you think they are, right? Because, right, yes, if there's just a single, like a transposable element in between these, then you can easily get a hotspot, right? Yes,
that's right. These are, and so the recombination hotspots are not determined by a single individual. These are phenomena that we see across multiple individuals. So unless there was some genome-wide perturbation in a subset of the population, which you could envision based on selection in the natural environment, it's unlikely that that is actually happening.
One last question.
You mentioned the juvenile to adult transition.
Yes.
So what confidence, or I'm sorry, that's a fair way to phrase it. To what extent can you be sure that these loci that you're pulling out from one-year-old trees, which I presume are still juvenile, will hold when they reach the adult or harvestable stage where you expect to use these trees as feedstocks?
So that's actually a very complicated question, although simply stated. because these are clonally propagated, you capture or you propagate their current developmental state. So we took cuttings off mature trees, clonally propagated those trees, and so they didn't rejuvenate. They didn't go back to their juvenile phase. So all the trees in the common gardens are of mature stature. The other side of your question is, does this data reflect what would happen when you commercially harvested these feedstocks for conversion to biofuels? And in most cases for poplar, the rotation ages are between age 5 and 8. And so we're not thinking about or talking about growing 20-year-old or 30-year-old feedstocks for conversion. So the short rotation narrows the range. As I mentioned, these studies are going to continue then. The data you see there is year 2. We're going to continue these up through year 5, which would be the commercial rotation age for an operation. Thank you.